So today's video is going to be a revision video on enzymes. As we are getting close to exams, I thought it would be useful to do some sort of revision videos to make sure that we are ready and prepared for exams. So today's video is on enzymes. So what do we know about enzymes? So the first thing that you guys should know, especially from GCSEs, is that enzymes are biological catalysts. So what does it mean by a biological catalyst, you may ask? So a catalyst speeds up the rate of reaction without being used up or changed. And it does this by basically providing an alternative reaction pathway for a chemical reaction to occur. In other words, it basically lowers the activation energy. So the activation energy is basically the minimum amount of energy needed for a reaction to occur. So if an enzyme is able to lower the activation energy, that means that more molecules will have energy equal to or more than the activation energy. Enzymes have a high turnover number. So a turnover number is the number of reactions an enzyme can catalyze per second. So that means that they can catalyze a lot of reactions in like 1 second, basically. Enzymes have an active site, and this is the site where a substrate actually binds to, and the active site has a particular type of shape. So that means that only a substrate that has a complementary shape to the active site can bind to it. So the active site and the substrate need to have a complementary shape. So not just any sort of substrate can bind to it. It must have a complementary shape. Complementary shape, you need to remember that because you could get asked a question on that in your exams, and complementary shape, well, that sort of phrase is something that examiners are looking for. And the active site, the shape of the active site is the tertiary structure of the protein, as enzymes are proteins, so it makes sense. And basically, it's just the shape of the tertiary structure that gives the active site its particular type of shape. And factors such as temperature, pH, and substrate concentration can affect an enzyme's activity, and that is something that I will be looking, well, it's going to be in the PowerPoint, so later on. So yeah. So there are two types of enzymes. You have intracellular and you have extracellular. So intracellular enzymes, you could be asked what an intracellular enzyme is. These are basically enzymes that work inside the cell. So an example of an intracellular enzyme is catalase. Catalase is an intracellular enzyme, and that breaks down hydrogen peroxide to water and oxygen. Extracellular enzymes, these are enzymes that work outside of the cell, so they are normally sort of secreted out of the cell. And examples of extracellular enzymes are amylase and trypsin. Trypsin breaks down proteins, so protease are enzymes that break down proteins, and amylase breaks down starch to maltose. Another thing you need to know about are cofactors. So some enzymes rely on small molecules called cofactors to catalyze a reaction. So a prosthetic group is a type of cofactor, and that is permanently bound to an enzyme. So carbonic anhydrase is an example of an enzyme, and its cofactor or its prosthetic group is a zinc ion, and that is permanently bound to the active site of the enzyme. So without that zinc ion, this enzyme, carbonic anhydrase, cannot catalyze a reaction. So carbonic anhydrase is found in red blood cells and is used to convert carbon dioxide and water to carbonic acid, and this later the carbonic acid later dissociates into hydrogen ions and hydrocarbonate ions. you need to know this when it comes to you learning about hemoglobin. What chapter is that? I think that's transporting animals. I think, yeah, that is in transporting animals. So you'll learn about that later on. It's also part of the Bohr effects and everything like that. You also have some cofactors that are temporarily bound to an enzyme's active site. So these cofactors may ease the formation of an enzyme-substrate complex, so basically making it easier to form an enzyme-substrate complex, and that increases the rate of enzyme catalyzed reactions. Some cofactors may bind to the substrate so they can form a complementary shape to the active site of the enzyme to form that enzyme substrate complex. Some cofactors change the charge distribution on the surface of the substrate molecule or on the surface of the enzyme's active site, and that basically makes it easier to form an enzyme substrate complex. An example of a cofactor that is temporarily bound to an enzyme is chloride ions, and these chloride ions are temporarily bound to an enzyme called amylase that I've mentioned, and that is an extracellular enzyme. Another thing you need to know about are coenzymes. So unlike cofactors, cofactors are inorganic molecules, whereas coenzymes are organic molecules, and these are temporarily bound to the enzyme's active site before or at the same time when a substrate binds to the active site. So these organic molecules are chemically changed and they are recycled. Coenzymes transfer chemical groups between enzymes, and most coenzymes are derived from vitamins. The lock and key hypothesis, it basically gives a sort of simple demonstration representation of how an enzyme catalyzes a reaction. So as you can see, you first have a substrate and you have the enzyme, and there I labeled the active site. So as you can see, the substrate has a complementary shape to the active site. So that means that the substrate can bind to the active site. It will literally fit perfectly. Once the substrate has bound to the active site, it then forms an enzyme substrate complex, as you can see. And then the enzyme catalyzes the reaction. One way it does this by destabilizing the bonds, like hydrogen bonds or maybe ionic forces within the substrate molecules. And then an enzyme product complex is formed. Then once the product is formed, the product is released, and that same enzyme can now find another substrate and bind to it again. So it is always being reused, it's not being used up, it's going to catalyze another reaction. Does that make sense? So now you have the induced fit hypothesis. The induced fit hypothesis is different to the lock and key hypothesis because the lock and key hypothesis shows that the active site has a rigid, fixed sort of shape, so it cannot be changed. Whereas the induced fit hypothesis shows that the active site can change shape a bit. It can change shape. So let's look at the diagram. So as you can see, just like the lock and key hypothesis, you have an enzyme, you have a substrate, and the substrate binds to the active site. However, you can see that the active site and the substrate, they have a sort of complementary shape, but it's not really as complementary, if that makes sense. So that means that when the substrate does bind to the active site to form an enzyme substrate complex, the active site changes shape just a little bit. This is just to sort of ensure sort of a tighter fit. So it kind of molds around the substrate so that it can get that tighter fit. And obviously the enzyme catalyzes the reaction by destabilizing the bonds in the substrate, and then an enzyme product complex is formed, same thing as the lock and key hypothesis. And then once the enzyme product complex is formed, the products are then released. And once the products are released, the active site changes its shape back to its original shape, to how it was before. So now we are looking at the effect of temperature on an enzyme's activity. So initially, an increase in temperature increases the rate of an enzyme catalyzed reaction. This is because the enzyme and the enzymes and the substrates have more kinetic energy, so they are moving around a lot. So that means that there are more successful collisions between the active site and the substrate. So that means more enzyme substrate complexes are formed. The rate of reaction increases up to the enzyme's optimum temperature, and this is the rate of reaction at its maximum, so at its highest, basically. Increasing the temperature past the optimum temperature, however, will cause the enzyme to denature. And this is because weak bonds such as the hydrogen bonds and the ionic forces that hold the tertiary structure of an enzyme together are broken. So that means that the active site changes shape. And if the active site changes shape, that means that the substrate can no longer bind to the active site of the enzyme because it no longer has that complementary shape. So the substrate no longer has a complementary shape to the active site. So fewer enzyme substrate complexes are formed. And that means that the rate of reaction also decreases. However, do not mistake this though. This primary structure is not changed, because the primary structure is it's the amino acids being bonded together. So the bonding of the amino acids is the bonds between the amino acids, the peptide bonds, are not broken. It is the hydrogen bond and the ionic forces bonds that are in the tertiary structure, that make up the tertiary structure. Those are the ones that are broken, not the peptide bonds. So make sure you know that. So this diagram right here basically shows what I basically said, that initially increase in temperature increases the rate of reaction all the way up to its optimum temperature. Then past its optimum temperature, the enzyme denatures, so that means that the active site changes shape, so that reduces the rate of reaction all the way down to 0, because the the active site no longer has a complementary shape to the substrate, so that means that fewer enzyme substrate complexes are formed. Another thing you need to know about is temperature coefficient. So you may be asked to calculate this in an exam. This is normally either a multiple choice question or is like a 1 or 2 mark question. So it is important for you guys to get those 1 or 2 marks, because those 1 or 2 marks can make a significant difference, to be honest, between an A and a B and an A star and an A, you know. Anyway, we're talking about temperature coefficient. So for a chemical reaction in a test tube, Q10 is approximately 2. So this kind of shows the sort of formula on how to work it out. So use a rate of reaction at temperature, 10 degrees higher than the actual temperature, divide by the rate of reaction at the temperature. There's not really a lot to say too much about this thing, but just remember, try and memorize and remember the formula and how to calculate it. So now we are looking at the effect of pH on an enzyme's activity. A low pH means that there is a high concentration of hydrogen ions, so an acidic solution basically. And a hydrogen ion has a positive charge, so it is attracted to negatively charged ions. This means the hydrogen ions are affecting the hydrogen bonds and the ionic forces between amino acids in the tertiary structure of an enzyme. A high concentration of hydrogen ions will disrupt the hydrogen bonds and ionic forces, so this will therefore alter or change the shape of the enzyme's active site, so then the enzyme is therefore denatured. Because it no longer has a complementary shape to the substrate, so fewer enzyme-substrate complexes are formed. However, not all enzymes have the same optimum pH of 7. Some enzymes have a low optimum pH. Pepsin, for an example, has a pH between 1 and 2, and that is generally because it is located, well, it works in the stomach, and the stomach has a low pH. Other enzymes may have a higher pH as well, and that could be possibly due to its location where it works. So don't always assume that an enzyme has an optimum pH of 7 because not all enzymes do. This basically shows a diagram of enzymes, of different types of enzymes. There are 3 different types of enzymes here, and as you can see, these enzymes have different optimum pHs. So not all enzymes have an optimum pH of 7, so some may be lower, some may be higher. So now we are looking at the effect of substrate concentration on an enzyme's activity. So initially, as the substrate concentration increases, the rate of reaction increases. And this is because there are more successful collisions between the active site of the enzyme and the substrate. So that means that more enzyme-substrate complexes are formed. And it also goes to show that substrate concentration is the limiting factor in the reaction. As you can see, the graph plateaus, so it curves off, and that basically means that after that, the rate of reaction stays constant, it's the same. And this is because all the enzymes are working at its maximum rate, the active sites are saturated or they are full up, something you can say. And it goes to show that substrate concentration is no longer the limiting factor. So there may be another factor that is limiting. Maybe the enzyme concentration is now the limiting factor. Yeah. And as you can see right here, there is a graph that you can look at and see. So the ones on the left hand side is basically explaining what the graph is saying, if that makes sense. Now we are looking at the effect of enzyme concentration on the rate of reaction. So this is going to be very, very similar to the effect of substrate concentration. So to be honest, it's pretty much the same thing if you ask me. But let's just kind of just talk about it. So initially, increasing the enzyme concentration increases the rate of reaction because more active sites are available, so more enzyme substrate complexes are being formed. This shows that the enzyme concentration is the limiting factor. If the substrate concentration is fixed, then the graph plateaus. As you can see, it curves off, so the rate of reaction stays constant, and this basically shows that the enzyme concentration is no longer the limiting factor. The initial rate of reaction. So the initial rate of reaction is the fastest part of the reaction. So at the start of the reaction, there are more successful collisions between the enzyme's active site and the substrate. So more enzyme substrate complexes are being formed. As the reaction proceeds and carries on, the number of substrate molecules available decreases as they are being used up and converted to the products. Therefore, the concentration of substrates decreases. So this means that the number of enzyme substrate complexes formed also decreases. So that means that the rate of reaction decreases. A lot of words, but it's something that you kind of have to know, and they can ask you in an exam, why do you measure the initial rate of reaction? We are nearly there, guys, so if you have reached this far, well done. We're nearly there. We've only got one more slide left, and then we're done. So competitive inhibitors. So a competitive inhibitor has a similar shape to the substrate, and by its name you can kind of guess that it binds to the active site of the enzyme. So it basically blocks the substrate from binding to the active site, and this means that fewer enzyme substrate complexes are formed. However, if the substrate concentration increases while the concentration of these competitive inhibitors stay constant, then the substrate molecules will outcompete the competitive inhibitor, as you can kind of see from the graph. So you can see that even with the normal sort of enzyme without the competitive inhibitor and with the competitive inhibitor, as the substrate concentration increases, they do meet up at a certain point. So that basically shows that the substrates are outcompeting the competitive inhibitors. Okay, so now we've got non-competitive inhibitors. So unlike competitive inhibitors, non-competitive inhibitors do not bind to the enzyme's active site. Non-competitive inhibitors bind to the allosteric site, which is away from the active site, so basically a different place. And when a non-competitive inhibitor binds to the allosteric site, it basically changes the shape of the active site. So that means that the active site no longer has a complementary shape to the substrate. So that means that fewer enzyme substrates are formed, and that basically reduces the rate of reaction. As you can see, when you compare the purple line with the blue line, which is the non-competitive inhibitor, you can see how the rate of reaction is lower than the normal one, and that is because the non-competitive inhibitor does not bind to the active site, it actually binds to an allosteric site, so a different site, and that alters or changes the shape of the enzyme's active site. Thank you for watching. I hope this was useful, and I hope this helped you with your revision, your biology revisions on enzymes. Hopefully I will do some more videos, like revision videos on biology. Maybe I could do some on chemistry, but that is not a sort of promise or a definite. So yeah, thank you for watching. If you want more videos like this, make sure to subscribe, like, comment, all those sort of stuff. And yeah, see you in another video. So bye guys, hope you guys have a good day. Bye.